Hey guys, welcome back to our lesson on population ecology. We just got done talking about different ways that we can estimate the size of plant populations and, of course, other organisms that are immobile. We talked about quadrats, transects, and different types of imaging technology. And ultimately, immobile organisms are pretty easy, all things considered, because, of course, they don't move. You can simply count them where they are. But animals, mobile animals, that is, get a little bit trickier. And so there are a lot of different ways to estimate animal populations as well. Um, we can simply listen and watch. Okay, so of course, if you see the animals, you can estimate the numbers based on a small area. And of course, this used to be a lot more difficult before photography was so good and so quick. But you would go out, and look at the sky and see all these birds, and you would pretty much just with your eyes section off a tiny little place like this, and as quickly as you possibly can, count as many moving birds as possible within that small little kind of aerial quadrat. And then again, just with your eye, estimate how many of those essentially quadrats are in this entire section. And so from scientist to scientist, from year to year, from like day to day, even, depending on how much coffee you had that day or whatever, light conditions, etc., estimates on populations in this way could be very, very inaccurate. And for the most part, they are underestimates of what the population size actually is. Since since digital photography has become so common and so easy, it is our estimates have improved quite considerably. So we can take a photo here, we can run it through technology such as ImageJ, and any little black speck is a bird. And so the computer can actually estimate pretty well how many birds are in this place. But what that means is that we can't compare current population estimates to population estimates from 20, 30 years ago, because they're measured in very different ways. We can use other signs, so vocalizations. This is really common in bird research. I've been on a few studies where, you just kind of like walk along a river, and every 50 meters, which you measure out by foot because you know your pace size, you listen for birds in that area, and try to detect the one that you're listening for. And so, of course, again, this is widely variable from person to person and day to day. You can look for things like scat. So scat, that is feces. I was on a study my senior year as an undergrad, in which we were looking at essentially wild rabbit or wild hare scat. And so, of course, they all look the same. And so what we did was we'd pull the scat into the lab, and we would chemically remove the outermost layer on the scat, which essentially contained the DNA of the rabbit itself. And we used PCR technology to determine what species of rabbit we were actually looking at, because, of course, rabbit scat looks like rabbit scat. You can look for tracks, right? Like deer tracks, if it's wolf tracks, etc. And another study that that I participated in as an undergrad was looking at bear fur. So essentially, we put out barbed wire, and so like as the bears would walk past, they would brush up against this barbed wire, and of course they have really thick fur. It doesn't hurt them, but it does pull out a few hairs, and so we would collect the fur to see what species, particularly of bear, was in that area. And so depending on how many individuals we found, and of course the range, that would help us to determine the size of the populations. Different types of surveillance. You can use like trail cams and see who is visiting the site. It of course takes a photo of whoever is around, and then you know that certain animals are there. Technology for locating animals has improved considerably, so much so that it is so tiny that you can actually have it on insects like this right here. There is a ratio of the animal size to the size and the weight uh, of the locator. You have to make sure that like bands on birds and these types of GPS technology, it has to be light enough that it doesn't actually inhibit the animal from doing what it needs to do. So these are really helpful, but not useful in all places. And of course, you can only tag so many animals. So, you know, estimating the whole population size from just a few has limited uses, limited applications. What I want to talk the most about is the mark recapture technique. And so in this technique, to estimate the size of a population, as many animals as possible are collected at a site, and they are marked in some way. So in this case, like the shell of a turtle is painted in some discrete place. On birds, you can like add a leg band. You can add a flipper tag on various marine species. So essentially, you mark every single animal that you collect, right? So in this example here, these scientists have collected 10 turtles and they have marked all 10 of them. Okay, so that's day one, right? So it might be like months or days or years or however long before the second collection day. And so when the scientists go out again to collect animals, they collect another number of turtles, and they are looking for how many of those turtles have the little paint mark. Okay, and so we can use these numbers in an equation. The number of animals captured first and marked from this first day sample, so these 10, all 10 of them were captured and marked. Okay, so that's a capital M. N is the total number captured the second time. And again, this can be hours later, it can be days later, it can be months or years later. It depends on the species being studied and the limitations of the scientists. But the total number captured the second time, which happens to be 10 as well, is lowercase n. Okay, and of the 10 turtles that were captured the second time, how many of them are marked? Right, and so this is R. Right? We call it R because these are essentially recaptured. So this guy was captured before, it has been recaptured, and we know it's been recaptured because it happens to have that little red paint mark. Okay, and so we can plug these numbers into an equation and we can get an estimate for N. And N is the estimated size of the population at this time. Okay, so we can use this number to predict later a lot of other things. Okay, so here we can see the total number captured in the second sample, the total number marked from the first sample, and the number recaptured. So let's take a look at the math for this. Here we see our variables have been defined. On day one, 10 were marked, that is capital M. On the second collection day, another 10 were captured, so lowercase n, and of those, 5 were recaptured, capital R. And so we are looking for the total number in the population, so n. Okay, so let's plug this in, lowercase n, the number captured the second time, times the number marked on the first day of the experiment over the number that were recaptured. So the number that were marked the second in the second collection day. And so 10 times 10 over 5 is 20 turtles total in the population. So let's try an example. Here, we're estimating the population density of Samuel song sparrow, which is this little guy right here. Nice brown circle here right in the front of the breast. And so this tells us that it's a song sparrow. And so initially, research team captures and bans 285 birds. They come back a second time and they capture 335 birds. Of those, only 4 were banned from the previous trip. And so in this question, it asks for what is M, what is N, what is R, and using these and the equation that we just looked at, what is the total population size to the best of our information that we have. So let's define our variables. On day 1, 285 were marked, capital M, that is, they were captured and banded. The second time, several months later, 335 were captured, so that's N, right? Total birds captured, and of those, only 4 were recaptured. That is, 4 of these initial 285 were recaptured. They still had the bands from the first capture. So that is capital R. So let's plug this in. We have N, that's what we're looking for. We want to know what is the population size of these song sparrows. Lowercase n times capital M over capital R gives us this many birds. Of course, we can't have 3/4 of a bird, so we can round up. 23,869 birds in this population, according to the mark recapture method. So on an exam, you should be prepared to calculate the population size based on information given from a mark recapture survey. And so to practice, I give you this example. So you're welcome to pause the video and try it out yourself. If you'd like the answer, look down in the comments section. So there are a variety of different indices that we can use to further estimate population. The Lincoln-Peterson Index of Relative Population Size uses a single mark, single recapture technique in a closed system. And so there are a lot of assumptions to actually using this index. And as you can imagine, there's a lot of ways that these assumptions can be violated, and therefore we couldn't use this particular index. So the assumptions are random sampling. So for example, as an undergrad, I went out with a group of biologists to band some birds at a nature preserve in Massachusetts. And so what we were doing was essentially using a net that was up in the forest, for hours at a time, and any birds flying by would get caught in this net. Carefully, the avian biologist would come along and remove the birds, put them in a little burlap sack so that they wouldn't be quite so stressed out. It was dark and they would kind of be subdued and calm, not so stressed out about the entire experience. The bird was brought back into this little field station here, cute little field station, and the bird was weighed. It was measured, like how big the wingspan was, how long the bill was, etc. And then, of course, any band that was on the leg, which maybe you can kind of see right here, was used in different mark recapture data sets. These birds, of course, were from this day, and this happens to be a golden crowned kinglet, so you can see just how tiny this guy is, and living in the frigid cold of New England. Anyway, this particular type of study would violate an assumption of the Lincoln-Peterson Index of Relative Population Size. The reason for that is that this chickadee, for example, now knows exactly where this net is, and it might remember and therefore be less inclined to go back to that same site the next time and get captured again. And so this would not be random sampling. This test happens to be sampling close to the field station. Okay, so random sampling is required in this single mark, single recapture system. Marked individuals must be randomly distributed through the population. And so similarly, if you're sampling in the same place over and over again, you might be catching the same bird over and over again, or the same turtle or whatever you're capturing. And so these guys might have a small range, and so you are more likely to catch birds that live in the local area, as opposed to birds from the entire population, right? So again, this assumption really difficult to meet. The ratio of marked to unmarked individuals shouldn't change between sampling periods. And so, again, really hard to meet all of these assumptions. A single mark, single recapture is very rarely used dependably in the field, especially for birds. And so there are additional methods which require multiple markings or multiple recapturings. We're not going to talk about them in any detail. I just want you to be aware that there are more methods that are a little bit more dependable. We are not going to be calculating anything else, just a simple mark recapture, which is essentially this Lincoln-Peterson index. And so once we estimate the approximate population size, we can use that number to estimate how the population will change over time. So will it grow, will it decline, and in what ways? And so the population size, again, measured by marking and recapturing, it changes when new animals are born. Okay, so new organisms coming into the population via birth. It loses populations via some organisms dying off. Okay, and two other factors here are immigration and emigration. Immigration is coming into the population, emigration is leaving the population. And so I have a few slides to introduce immigration and emigration to you, and maybe some different circumstances where this would be the case. But as far as our math goes, we are only going to be looking at the population size and how it changes according to births and deaths. Okay, so yes, these are super important. We're going to talk about these circumstances, but we're only calculating down here. Okay, so our first examples are going to use these equations, and then we'll kind of go out from there. So maybe taking note of these, maybe making up a big sheet of all of these equations would be a great way to reference while you're doing homework assignments, as well as when you are taking an exam. Okay, so let's start. A wildlife preserve is the home of a starting population of 100 tigers. And so the starting population, we would call this, the population N, capital N, in year 0, right? So the very beginning. The following year, ecologists note that 40 cubs were born and 10 tigers died. Okay, so the following year, we call this year 1. And in that year, 40 more cubs were born on top of the existing 100, and 10 of the tigers died. Okay, given this trend, what is the expected population in the next year, in year 2? And so, of course, we could very easily add these to 100, subtract the 10, and calculate that, okay, it's 130 tigers in year 1. But we have enough information using these equations down here to predict what the population is going to be in year 2. So let's give it a shot. In this problem, it will be important to define what population we're talking about, that is, in which year. And so we'll be using N, right, for the total population size in year T. Okay, so population in year T, because we start out in 1 year, we measure again in the next year, and then we want to know about the year after that. Okay, so NT, this is the year we're talking about. And so the problem tells us that N0, right, the starting population, is 100 tigers. We could simply calculate how many tigers there are in the population in year 1. Right, so the next time, these scientists go back in year 1 with this equation. Right, so what is the population in the starting year, N0, plus how many tigers were born, minus how many tigers died, plus, of course, how many immigrated in, right? So this would probably be negligible because it's a preserve and, it might be fenced off, and minus emigrants, so tigers that are leaving. And again, this is unlikely because of the fenced-in nature of the preserve. Okay, so again, if we want to calculate N1, right, so T plus 1, 0 plus 1, we can add the births, subtract the deaths. We know from the problem that the number of births is 40 and the number of deaths is 10. And so we use capital B for the number of births, right? So like the number of animals itself, capital B, the number of deaths, 10 tigers died. Okay. So again, we can do that calculation. Okay. However, in order to predict what the likely population is going to be in the next year, right? So we haven't measured this, but given this trend, so 100 tigers to 130 tigers in N1, right, year one, we need to calculate how quickly the population is growing. So this is actually a rate. It's both number of organisms and time. So let us calculate the birth rate and the death rate. Okay? The birth rate is expressed as lowercase b for the rate, and capital B for the number of births. Okay, so in this equation, we can see the birth rate is the number of births, 40, over the number of animals, so 100 tigers. Okay, so 40 over 100 is 0.4. This is the birth rate. The death rate is expressed as lowercase m. And so lowercase d is something different, and we'll look at DNDT and all sorts of things, but this is essentially meaning mortality. What is the mortality rate? And so the number of deaths, 10, over the number in the population, 100. 10 over 100 is 0.1. So now we have the birth rate and the death rate. We can calculate the population growth rate with this equation. Okay, growth rate is R. And essentially, if this is positive, it's meaning it indicates that the population is growing. If it is negative, it implies that the population is declining. Okay, so to calculate R, the birth rate 0.4 minus the death rate 0.1 gives us a 0.3. So this is positive. It means that the tiger population is growing. Okay. So given the birth rate, the death rate, and the per capita growth rate, we can calculate the estimated population size for the next year. And we can do that in two different ways. I'm going to show you the longer way that we're not going to do again, but hopefully you can kind of see how all of this stuff comes together. And then I'll show you the way that we are going to do it for real. All right, so option one, we can calculate the number of animals in the population in year T, whatever year that is, whether it's year 1, year 2, year 17, whatever. We can do that by taking the previous year's population size, T minus 1, okay, number of animals last year, plus the number of animals last year times the birth rate. Right? This gives us how many tigers were born, minus the number of animals last year times the mortality rate. This gives us how many animals died. And so this is essentially like what we've done previously, 100 tigers plus 40 minus 10. It is exactly this here. And so just to replace the T minus 1 here, T is 2. So we're curious about tiger population in year 2. Last year's population, year 1. How many animals were born from last year? How many animals died from last year? So if you plug in these numbers, what we see is 130 animals, which we calculated here, plus the number that were born minus the number that died. And what we get is 169 tigers are expected in year 2. Okay, so in order to calculate year 3, you would need to know year 2, and so on and so forth. So if you were curious about year 10, you would have to calculate every single year so that you always had n T minus 1. Okay, so that's the long way. Of course, the way that maybe hopefully makes sense, you can logically walk yourself through that. We are going to try another way. We are going to use this equation. So nt plus 1, what is next year's population size, equals lambda, which is called the finite rate of increase, where lambda equals 1 plus the per capita growth rate, okay, times this year's population. So for us, 1 plus 0.3 times 130, right? This is the population in year 1. Multiply together, and we get the same answer, 169 tigers in year 2. So again, this is just 2 different ways of calculating the possible population in the next year. For both of these, you have to calculate every single year. So if you wanted population in year 3, you would need this number. If you wanted the population in year 4, you would need the next number that you calculated for year 3. As I said, the only math that I'm going to have you do is going to use birth rates and death rates. It is not going to be looking at immigration and emigration. But it is important to know that those are 2 very important factors in determining population size. Ultimately, in many species, individuals move within the population. And one way that happens is called dispersal. So immigration, emigration usually implies moving away from each other. And so this can be done in a variety of organisms. For plants, for example, seeds can be dispersed. Most of them fall near the parent plant. And so what this image is showing you are 1, 2, 3, and 4 seed trees. And I know it looks like a topographical map, but essentially each one of these rings is showing you 100,000 fewer seeds are dispersed here. Right, so as we can see, like 500,000 seeds are dispersed right around these 2 seed trees, but the farther and farther away that you get, the fewer and fewer seeds are coming from these trees. And so this dispersal is kind of like emigration. Depending on the species, this will determine just how far the seeds actually go. Wind carries the seeds that are light. Heavier seeds, of course, have a shorter dispersal distance. Okay, so population dispersal is affected by seed size and weight. Animals can also disperse seeds. Okay, so depending on the type of seed, depending on whether it has a fruit around it, depending on whether it has like barbs that catch into animals' fur, these factors determine how far the new individuals are going to be dispersed. Animals also disperse. Of course, you can think of like Charlotte's Web at the end there, where all like Charlotte's baby little spiders are like blowing away in the wind. That's a real thing. So spiders can blow away. Water carries larvae. And actually it carries a lot of organisms away into new environments. Animals can actively disperse. So juvenile birds, juvenile mammals even, move into new habitat, particularly in territorial animals. So the young can't inhabit the same territory as the parent after a certain point, and so it must go out and find a new place to live. And of course, dispersal is very much impacted just by what's available, right? So if the place is really crowded,